In this video, we are going to look at protein denaturation, causes and the effects. Protein denaturation refers to the loss of native conformation of a protein. On denaturation, a protein loses its physical, chemical and biological properties. So on the board I have illustrated an example of a protein. So this is the native conformation of a protein. That is, it is highly folded. So on denaturation, our protein is going to lose its native conformation, like that. So the causes of denaturation are as follows: acids or bases. So strong acids or bases normally interfere with the native conformation of a protein by cleaving the peptide and sulfide bonds. Another cause of protein denaturation is heating our protein to a temperature of between 50 and 80 degrees Celsius. So when you heat a protein to such a temperature, you denature the protein, you destroy the tertiary structure of the protein. So detergents and organic solvents are hydrophobic in nature. This hydrophobic structure of our detergents interact with the hydrophobic side chains of proteins. So in the process, this causes disruption of the tertiary structure of a given protein. Examples of organic solvents that disrupt the structure of a protein are alcohol and acetone. Another cause of protein denaturation is heavy metals. Heavy metals such as cadmium, lead, and mercury interact with sulfhydryl groups and also carboxylate groups of a given protein. The affinity of heavy metals to functional groups in proteins make them highly toxic to proteins. That's why in a hospital, a patient who has been poisoned or who has taken poison is first of all treated with an egg. So the proteins in the egg will interact with the heavy metal ions. Remember that poisons contain heavy metal ions. So that means that will render the poison harmless. Other causes of denaturation include the use of urea and quinidine at a high concentration. Also, the sonication of a protein and also vigorous shaking can cause denaturation of a protein. High pressure, UV rays and X-rays can also cause protein denaturation. That is a very important information for you also to note. Please note that protein denaturation does not affect the primary structure of a protein. It's also important for you to note that on denaturation, a protein may be renatured, but not all proteins can be renatured on denaturation. Another thing is denaturation of protein decreases solubility and increases the precipitation of a protein. So it increases solubility and decreases precipitability of a given protein. So let's go through our notes. Protein denaturation refers to the loss of native conformation of a protein. Denaturation alters physical, chemical, and biological properties of a protein. Causes of protein denaturation. Number one is heating with strong acids or bases. This cleaves peptide and disulfide bonds. Heating above a certain temperature, that is 50 degrees Celsius and 80 degrees Celsius, it destroys the tertiary structure of proteins. The process is called heat denaturation. Then another cause is detergents and organic solvents. Disrupt hydrophobic interactions. Being nonpolar, they insert their effects between side chains of hydrophobic amino acids. Examples of such organic solvents include ethanol and acetone. Another cause is heavy metal ions. Lead, cadmium, and mercury can denature proteins by binding to their carboxylate and sulfhydryl groups in proteins. The affinity for functional groups makes heavy metals toxic to proteins. Other causes: use of urea or quinidine at a high concentration, sonication and vigorous shaking, high pressure, UV rays, and also X-rays. And B, the primary structure of a protein is not altered during protein denaturation. Denaturation reduces solubility and increases precipitability. Denatured proteins are sometimes renatured when physical agents are withdrawn. Ribonuclease is such an example. However, many proteins undergo irreversible denaturation. Kindly like, share and subscribe to my channel.